So this will be the part 2 of antimicrobial agents, and in this class we will discuss about antimicrobial susceptibility testing or AST. This is not a big topic and is very simple, and on exam point of view we can expect the whole antimicrobial susceptibility testing as a short note question. Now, what is susceptibility? Susceptibility of a pathogenic bacteria is to be determined because it is the liability of the bacteria to be influenced or harmed by anything. Here, what we harm the bacteria is by the antibiotic. So we have to understand how much the antibiotic can harm the bacteria, and according to that we will select our treatment methods. So this is why we are performing antimicrobial susceptibility testings. So it is performed for bacteria which are isolated from the specimen and not for commensal bacteria. That is, commensal bacteria like which are already present in the flora of our body. We will not perform tests for those bacteria. For example, E. coli is isolated from the urine specimen. We will do the AST for the urine specimens E. coli, but we will not do it for the stool because it is a commensal one. E. coli is normally present in the stool, so we will not perform an AST for E. coli in the stool, but it is done for E. coli in the urine specimen. So next we will go to the classification of ASTs. So it is classified as disk diffusion methods, dilution test, E-test, automated methods and molecular methods. Now we will go into each of this and go into the explanations. First one is disk diffusion method. So this one is the most widely used and suitable one for rapidly growing bacteria, and therefore it is unsuitable for the slowly growing bacteria. We will use another method for them. This is for the rapid growing bacteria, and it is named so because we uses the filter paper disc impregnated with appropriate concentration of the antibiotic solution. This is why we name it as the disk diffusion method. Now what we do in this method, that is, here the test bacterium is isolated and we inoculate it on a solid medium, and then the antibiotic discs are applied. So you must have studied what are the culture mediums like solid medium, liquid medium, everything we have studied in the previous classes. So here we inoculate it into a solid medium, and then the antibiotic solution diffuses through the solid medium so that the concentration will be higher near the site of the application and lower away from the site of application. Now we have to determine the sensitivity to the drug, and it is determined by the zone of inhibition of bacterial growth around the disc. So there will be a zone where it is inhibited from growth, and according to this we will be determining the sensitivity. Next we go on to the medium, and the medium used here is called as Muller-Hinton-Agar. That is Muller-Hinton-Agar. So this is the best medium for growth of non-fastidious pathogens, and it is best for them since it has a minimal inhibitory effect on sulfonamide and trimethoprim, and it will also support satisfactory growth of most of the non-fastidious pathogens. So we are using this medium other than any other mediums. So since we are using them for non-fastidious pathogens, we have some modifications of MHA also, like lysed horse blood added to the MHA for growth of fastidious organisms like Haemophilus influenzae. We have said that this is the best medium for non-fastidious pathogens. We can also use it for fastidious organisms like Haemophilus influenzae when we are modifying the MHA. And we have also another modification, that is sodium chloride. When it is added about 2 to 4%, we can use it for testing the MRSA isolates. And MRSA is methicillin resistant Staphylococcus aureus. So next we go on to the inoculum. So the isolated pure colonies of the test organism, we will inoculate it in a suitable liquid medium like peptone water broth, and we incubate at about 35 to 37 degree Celsius for about 4 to 6 hours. So we will compare the turbidity with that of 0.5 McFarland opacity standard 2. So it is a standard turbidity, and we will be comparing with that one and setting the density. So then the inoculum is done with the lawn culture. The broth will be then inoculated on the medium by spreading with sterile swabs. Now, what will be an ideal inoculum? An ideal inoculum will be after overnight incubation, it will give an even semi-confluent growth. So if we keep it for more time, it may become heavy, and the too heavy inoculum reduces the size of inhibition zone. So we have said before that is with the inhibition zone we are calculating the susceptibility. So we have to do it perfectly for producing an ideal inoculum. Next is about the control strains. Similar to the test inoculate, we have to also include control strains for testing AST. And the ATCC strains, or that is the American type culture collection. We are using standard control strains like E. coli, Pseudomonas aeruginosa, Staphylococcus aureus, Enterococcus faecalis. So these are used as the standard control strains according to ATCC. Now to the antibiotic disc. Antibiotic discs are available commercially or also prepared in the houses. So like the Wattman #1 filter paper, they are sterile filter paper, disc of 6 mm diameter impregnated with antibiotic solution is available. Now about the choice of antibiotic disc. It is neither possible to test all the drugs again for susceptibility. So the panel of drugs to be tested against the isolate depends on certain factors, and they are the antibiotics should likely to be used for the therapy. We should test for the antibiotics that may be used for the therapy. And another factor is the organism against which the drug is to be tested, and the local prescribing habits of the antimicrobial agents, it is also a factor, and the resistant patterns of the locally prevalent pathogens. So the normal resistant pattern of the pathogen is a considerable factor, and also the cost, toxicity, pharmacokinetics, and spectrum of activity of the antimicrobial agent for the management of the illness in a particular patient. For each patient it will be different, so we have to consider each of these factors and do the antibiotic susceptibility test. Now we do the testing for both first line and second line drugs. So what are the first line drugs? Those are the antibiotics that are commonly used in the locality with respect to the organism isolated. So these should be tested first, and they are the first line drugs. And here we should also include the antibiotic currently administered to the patient. If the patient is taking any other antibiotic, we should also test that one as a first line drug. So here the spectrum of the antibiotic should be relevant. Next is second line drugs, and it will include a panel of antibiotics for which prescription is restricted only to special circumstances. So we will test this only if the organism is found resistant to all the first line drugs. We have to test it for a second line of drugs also. Now we will go on to each of the methods, and first one is Skippy disc diffusion method. So here what we do is, first a cotton swab is dipped into the inoculum, and then we will squeeze it and drain out all the excess fluid. Then the swab we will inoculate it into the Mullen-Hunton agar, which we said before, by streaking the swab 3 times over the entire agar surface. Then we will keep it to dry, and after it is dried about for 3 to 5 minutes, we will apply the antibiotic discs on it. And we have to note the disc should not be placed closer than 20 mm onto the MHA plate. So usually about 6 discs are placed, 5 around the periphery and 1 in the center. The plates will be then incubated at normal body temperature or 37 degrees Celsius for about 16 to 18 hours. Then the zones of complete growth of inhibition around each of the discs are measured using a vernier caliper or a ruler, and this diameter is included in the measurement. Now each of the zone we will interpret it into 3 parts, that is the sensitive, intermediate or resistance, according to which we will be testing it. This is the image of Kirby-Boer disk diffusion method, and this is the arrangement. Here you can see the disks are arranged in the periphery and the zones of inhibition are marked. Next one is the Stokes disk diffusion method. Here the MHA plate will be divided into three parts. The test organism is inoculated on the central one-third and the control strain on upper and lower thirds of the plate. So in modified Stokes method there will be a change, and maybe the test bacterium will be placed in the upper and lower part also. So there will be a gap, an uninoculated gap of 2 to 3 millimeter wide in between the test and the control area, in which the antibiotic discs will be applied. And the plates are then incubated at 37 degrees Celsius for 16 to 18 hours. Now is the reporting. It is the interpretation, and it is given by comparing the zones of inhibition of control and the test bacterium. So here we will be measuring the radius from the edge of the disc to the edge of the zone. We interpret it as 3, so that is sensitive, intermediate and resistant. The radius is wider than or equal to or not greater than 3 mm smaller than that of the control. And the intermediate is where the radius of the test will be greater than 2 mm but smaller than the control by a part of greater than 3 mm. And the resistant is where there will be no zone of inhibition for the test, or the zone radius is 2 mm or less than the control. Next is the primary disc diffusion test. It may be performed when the results are required urgently and a single pathogenic bacterium is suspected. And here the clinical specimen is directly inoculated uniformly on the surface of the agar plate and the discs are applied. So this test is of no use when the mixed growth of different bacteria is suspected to be there in the specimen, specimens like the pus, stool and sputum. There may be mixed growth of different bacteria. So here we can't do this method. When we are suspecting the pathogenic bacteria as a single one, we can do this test. So the next classification is dilution test. Here the antimicrobial agent is serially diluted and each dilution tested with the test organism for AST, and we calculate MIC. Now what is MIC? It is a minimum inhibitory concentration, that is the lowest concentration of an antimicrobial agent that will inhibit the visible growth of the microorganism after an overnight incubation. So depending upon whether the dilution of the antimicrobial agent are made in agar or broth, we will classify it into two types of dilution test. That is the broth dilution method and the agar dilution method. So before going into classification, we will say about the importance of MIC. So it is also useful for confirming the results of AST, and we use it for testing the antimicrobial sensitivities of the slow growing bacteria like tubercle bacilli and bacteria for which the diffusion test is not standardized. Here also we will perform MIC, and it is useful to demonstrate very small degree of the resistances and when therapeutic dose of drug has to be regulated. So in all these cases MIC calculation is important. What is broth dilution method? Here we perform serial dilutions of the antimicrobial agent in the Muller-Hunton broth and are taken in tubes, and each tube will be inoculated with a fixed amount of suspension of the test organism. We should also include a control organism whose sensitivity is already known. And now we will incubate it at 37 degrees Celsius for 18 hours. Now we will determine the MIC by noting the lowest concentration of the drug at which there is no visible growth, that is the broth appears clear. So when the broth appears clear, it is at that point we will understand there is no growth of the bacteria now, and here we will calculate the MIC. So the tube containing the lowest concentration of the drug that fails to show growth on the subculture is the MBC of the drug, and MBC is the minimum bactericidal concentration. How we obtain it? It is obtained by the subculturing from each tube showing no growth. So there will be some tubes where there is no growth of the bacteria. From that we will subculture a little amount or a little concentration onto a nutrient agar plate without any antimicrobial agent. From this one we will calculate the minimum bactericidal concentration. So this image shows the broth dilution method. On the right end we can see the control, and in other test tubes we can see that the turbidity is indicating the growth, and on the left side we can see they are the subcultured ones into the nutrient plate. and after subculturing we found there are some no growth and growth, and from this no growth we are calculating the MBC. Next one is the Agar dilution method, and this method is more convenient than the broth dilution method. Why? First, how it is done. Here the serial dilutions of the drugs are performed in molten agar and poured into petri dishes. Then the test strain will be inoculated. So why this is more convenient is that it has an added advantage that several strains can be tested at the same time by the same plate. There is no need of changing each of the plates every time. And next, it directly measures the MBC. There is no need of subculturing into another nutrient medium. Next classification is the Epsilometer or E-test. This is a quantitative method of detecting MIC by using the principles of both dilution and diffusion of antibiotic into the medium. So it uses an absorbent strip containing predefined gradient of the antibiotic concentration immobilized along its length. So the next page will show the figure of Epsilometer, and we can see the gradient very clearly. And here it is then applied to the lawn inoculum of the bacterium following incubation on the elliptical zone of the inhibition produced. The antibiotic concentration at which ellipse edge intersects the strip is called as the MIC value in E-test. So here we can see the figure. In this figure you can see that the elliptical edge which is intersecting the strip is calculated as the MIC. So the final classification is molecular methods. There are available PCR based assays for targeting the specific drug resistant genes. We know the molecular assays will be acting at the molecular genetic level, and it is very suitable for the detection of MECA gene for the MRSA detection. So that's all about the topic. We should have a basic awareness of all the antimicrobial susceptibility testing, and it is the least expected question, but we can expect it as a short note question. That's all. Thank you.